Thank you so much for inviting me. And I'm super honored and also very excited to share my work with everyone here today. I'm a fourth year PhD student in Dr. Jill Banfield's lab at UC Berkeley. For my talk today, I'm going to share with you my journey of looking for contaminants in metagenomics dataset. Before I go into the detail, I just want to give you a brief background on how I got into this contamination project. So I was working on this project trying to understand early life gut microbiome succession at the strain level. Specifically, I was curious to learn how babies share strains as they grew up. I also wanted to look for persisting strains that were able to basically be there from the birth until baby turned age 1. And then lastly, I wanted to kind of understand how maternal strain transmission play a role in the early life strain succession. So in total, we collected over 400 samples for this project. But in the end, we were only able to do analyses on half of the samples. And that is because the other half of the samples were suspected to be cross-contaminated. So how do we discover cross-sample contamination? So now I'll walk you through the contamination detection framework that we devised. And I really hope this workflow can be a useful resource for the microbiome field in general. So for the infant gut project, since we had over 400 samples, we chose 96 well plates for DNA extraction just because it's much in higher throughput than single tube extraction. And then for each extraction plate, we included one reagent-only negative control, and then we named them based on the plate number. So here you're seeing NC1, refer to negative control 1 in the plate 1. And to avoid cross-plate contamination, none of the plates were extracted at the same time. So when there's no contamination, you would not expect to see any organisms in these negative controls. And that was the case with NC1 and also NC5. However, we saw at least one organisms in NC2, NC3, and NC4. So here I'm showing you the taxonomy breakdown of the genomes that were detected in these 3 negative controls. And these genomes were colored by phylum. And these black lines represent individual genomes within each corresponding phylum. So as you can see, while NC2 only had 1 genome, NC3 and NC4 had about 60 bacterial genomes. So here I'm showing you a closer look. So I'm plotting the top 8 most abundant organisms. And just by looking at the species name, it seems like the majority of the genomes that were detected in NC3 and NC4 look like common gut microbes. So to kind of look for the sources of contamination that were detected in negative controls, we decided to look for near identical strains that were shared between fecal samples that were extracted from this project and also all the negative controls. And to look for near identical strains, we used a recently published tool from our lab called InStrain. So when determining whether 2 samples share the same or different strain, InStrain looked for population-wide allele differences across the genome, therefore considering both the major and also minor alleles for all positions instead of just consensus alleles alone. So once we found all shared strains between fecal samples and negative controls, we visualized such strain sharing pattern in the context of DNA extraction plates. And this is what we saw. So P1, P2, P5, all the negative controls on these 3 plates never shared any strains with any fecal samples. However, NC3 and NC4 only shared strains with samples extracted from the same plate. And as you can see, there is some sort of proximity-based strain sharing pattern going on. So I want to zoom in to just talk about one plate so you understand how to read such plot. So all the circle here represent samples. And if a fecal sample shared strains with the negative controls, a line would be drawn between them. And then the more strains the sample shared with the negative controls, the brighter and the thicker the line gets. So in this case, the sample right here shared the most number of strains with NC4. So after seeing this data, we were wondering, and also worried at the time, whether there were any other fecal samples were cross-contaminated. So to answer that, we looked for strain sharing among all sample pairs from unrelated infants. And then we visualized such strain sharing pattern in the context of DNA extraction plates. And this is what we saw. So P3 and P4 look quite busy, while P1, P2, and P5 look quite clean. And I want you to pay attention to these circles that are colored pink. These are the samples that displayed location-based and or plate-based strain sharing pattern. This sample right here, it is not sharing any strains with any other samples that were extracted from other 4 plates. However, it is sharing strains with at least half of the samples on this P4, suggesting that there might be some plate-wide spillover event happening to this sample. And then we confirmed that these samples were indeed cross-contaminated by re-extracted and resequencing these samples. And when you plot such re-extracted samples on the plates, this location-based sharing patterns were not seen anymore. So we conclude that this data set, for this data set, the major source of contamination is internal contamination, specifically wall-to-wall contamination. And given how important strain-resolved analyses were for this early life strain succession project, we made a conservative approach by discarding all samples that were extracted on these 2 plates and move forward our analyses with samples on these 3 plates. And also with this kind of strain-resolved workflow, we were wondering how applicable such workflow is for other metagenomics data set. So we apply this kind of workflow to a different preterm infant data set, and this data set is not published. So for this data set, we had samples from birth until month 2. And besides fecal samples, this data set also had tongue swabs and chest swabs. And to look for the existence of contamination, we started by also looking at reagent-only negative controls first. And as you can see, there were at least 1 organisms to be detected in these 3 sequenced negative controls. We then look for unusual strain sharing patterns in the context of DNA extraction plates. And in the end, we concluded that unlike the first dataset, this dataset was primarily contaminated by externally derived contaminants. And these contaminants included the ones that I'm showing you here, which were detected in the negative controls. So to conclude, contamination can be insidious from sample collection to DNA extraction to sequencing. Contamination can occur anywhere along this pipeline. So it is very important to assess whether the organisms in front of you are truly belonging to the samples or not. With the help of negative controls, you're able to detect and also remove organisms that were accidentally introduced prior to sequencing. And also with negative controls, you're able to kind of be aware whether there's a existence of within sample contamination, such as wall-to-wall contamination. However, looking at negative controls alone is insufficient when assessing contamination. So it is important to look beyond the negative controls to look for any artificial strain sharing patterns among samples from unrelated groups, as the ones that I showed you here today. So I hope this work has brought your attention to contamination, especially contamination that originated within the study, such as wall-to-wall contamination. I think it is a topic that we don't talk enough, but can play a very significant role in microbiome data interpretations. So I just want to end by thanking my lab, especially my PI, Dr. Jill Banfield. And also I want to thank our collaborator from University of Pittsburgh. With that, thank you for listening, and I'll see you at the Q&A.